How to get beer cheaper. What improves the efficiency of yeast fermentation? What is fermentation? Well, it's a metabolic process that takes place in the cytoplasm of yeast cells that converts sugars into CO2 and ethanol with no oxygen present. This is through glycolysis, which breaks down glucose into pruvates. Those pruvates then allow for production of a small amount of ATP. As well, those pruvates are converted into ethanol and CO2, which hence they're not joining the citrus acid cycle. This process then allows for the regeneration of NAD, therefore continuing the glycolysis cycle, which is essential for that cell to survive. Why does does the experiment matter? Well, the production of ethanol through fermentation of yeast is used by many billion-dollar industries such as the production of fuel, pharmaceutical, and food and liquor. This experiment aids in determining and understanding which sugar substrates ferment most efficiently, allowing for a deeper understanding of yeast metabolism. Hence, those industries can use the knowledge to improve their own fermentation processes, which allows for a more optimal production of ethanol, which cuts costs and creates more efficient renewable energy, as ethanol is a major focus in renewable energy studies. The overview of this experiment. To achieve this, we used different substrates which were tested against a buffer solution. We warmed each substrate, then the mixture was placed in Durham vials. Next, the substrate were inverted in the test tube so fermentation occurred. In those Durham vials, we were able to measure the CO2 collected at the top, which we measured over 45 minutes. The fermentation produced both CO2 and ethanol, hence the CO2 production is directly proportional to the ethanol formation, allowing us to measure the rate of production of ethanol through the fermentation. In figure 1, it shows the production of CO2 over 45 minutes recorded in 5-minute increments of the different substrates we used. We see that the yeast and corn syrup substrate had the highest production of CO2. In figure 2, it shows the results of the linear rate of CO2 production. It was calculated using the log phase of figure 1, where the slopes of the linear parts were calculated. We see that as well, yeast and corn syrup having the highest production of CO2. Discussion. This experiment demonstrates that yeast and corn syrup yielded the most and highest rate of CO2 production. Our results seem to align with the basis of fermentation needs, like the simple sugars, which would enhance the production of glycolysis. Yeast and corn syrup had the highest fermentation rates due to the increased availability of glucose. Hence, glycolysis can use that immediate availability of glucose to quickly start the fermentation process. The kernel and cellulose and yeast treatment had the highest production of CO2, or had a higher production of CO2 than the kernel and yeast reinforcement, than the kernel and yeast treatment, reinforcing the conclusion that enzymatic hydrolysis occurs due to the added cellulase, which increases the availability of sugars for fermentation, hence allowing for a more productive fermentation. While the kernels and cellulase with no yeast had the lowest production of CO2 produced, which demonstrates that the enzymatic hydrolysis alone does not increase the CO2 production, hence showing that the yeast is very essential in fermentation. The water and yeast had a lower production rate than just the yeast and kernels, which aligns with the need of an appropriate sugar substrate for productive fermentation. In conclusion, this experiment provided findings around the most efficient fermentation. This experiment provided findings around how the most efficient fermentation requires high amounts of accessible simple sugars. The cellulase was found to increase the fermentation efficiency with the kernels. These findings can aid industries using fermentation to produce many widely used products to enhance their fermentation processes by incorporating cellulase and substrates with increased amounts of sugar to improve glycolysis. Future research should really focus on the addition of cellulase to bio-waste, which could be fermented into ethanol, which would be used for fuels, allowing for an increase in waste reduction, pollution, and a lower production cost. My final words I would like to thank Jack Yang and Janelle McCain for their help with this assignment, and thank you for your time.